Hello everyone, welcome back to my YouTube channel Shala by Dr. Rohit Sharma. Today we are going to discuss this topic apoptosis. So what is apoptosis? A literal meaning of apoptosis is falling off. It is also a form of cell death like necrosis where cells activates intrinsic enzymes and these enzymes are going to degrade own nuclear DNA as well as nuclear and cytoplasmic proteins. As you can see in this image, this is a normal cell and these are the falling off bodies. Now this is a beautiful image showing necrosis on the left side and apoptosis on the right side. As you can see in this image on the left side, the plasma membrane gets disrupted and cytoplasmic enzymes leak out of the cell to cause the ultimate damage. Whereas in this image on the right side, this cell is transforming into multiple bodies and these bodies are known as apoptotic bodies. Now moving on to the causes of apoptosis, we have some physiological causes as well as pathological causes. In physiological causes, we have embryogenesis, we have hormone withdrawal, example is endometrial breakdown, and which hormone is going to play an important role in this is progesterone, because progesterone is the hormone that maintains the endometrial lining. Whenever there is fall of progesterone levels, this endometrium lining is going to shed off by apoptosis. Then elimination of self-reactive lymphocytes. These self-reactive lymphocytes are going to react with the self-antigens and cause autoimmune disorders. That is why they need to be eliminated by apoptosis only. And we have death of neutrophils after healing infection. In pathological causes, we have DNA damage that is beyond repair. We have accumulation of misfolded proteins and we have viruses that may cause apoptosis. Now let's talk about the biochemistry of apoptosis. What happens in apoptosis? There are some procaspases which are in inactive form in our body. These procaspases are going to be converted into caspases and these caspases will activate endonucleases. Now, as the name suggests, these endonucleases are going to cause chromatin condensation, and secondly, these endonucleases are going to cause nuclear DNA damage. But this nuclear DNA damage is going to happen at a specific fragment, and this specific fragment is between the nucleosomes. That is why it is known as internucleosomal DNA damage. That is a very important MCQ that has been asked many a times in NEET PG and AIIMS. Now let's talk about the mechanism of apoptosis. Apoptosis happens in two phases. One is the initiation phase, another one is execution phase. This initiation phase can occur with two pathways. One is intrinsic pathway, the second one is extrinsic pathway. And as you can see that I have written intrinsic with a number 9 over here. What does it signify? That caspase 9 is going to function in intrinsic pathway. That's how you can remember it. That intrinsic has caspase 9. And in extrinsic we have caspase 8 in lower animals and caspase 10 in humans. In execution phase we have caspase 3, 6 and 7. They are going to activate endonucleases, and at the end of apoptosis these endonucleases will cause chromatin condensation. So this is the whole mechanism of apoptosis. In brief, we are going to discuss it in details as well about intrinsic pathway and extrinsic pathway. Now, before going to the detailed mechanism of apoptosis, I just want to tell you about some proto-oncogenes. In proto-oncogenes, we have a few genes which are pro-apoptotic and few genes are anti-apoptotic. In pro-apoptotic genes, we have BAC and BAX. In anti-apoptotic genes, we have BCL2, BCLXL and MCL1. So many a times MCQs have been asked, example, all of the following are anti-apoptotic genes except. The options will be BCL2, BCLXL, MCL1 and BAC. So what are you going to mark? The answer will be BAC. Likewise, we have some sensor they are present over the cell surface and they are also known as BH3 only proteins. In sensors, we have BIM, BAD and BID and Puma and Noxa. How can you remember this? With the mnemonic that BIMLA is bad at bidding. That's how you can remember it. BIM, BAD and BID. Now something about intrinsic pathway in detail. This pathway is also known as mitochondrial pathway and it is a major pathway where mitochondria plays a very important role. Suppose this is a cell, we have mitochondria inside the cell and this mitochondria is comprising of two membranes. One is outer membrane, the other one is the inner membrane. And as you can see, this inner membrane is thrown into folds. What are these folds known as? They are called as cristae. On the outer membrane, we have BCL2 that is anti-apoptotic gene. On the inner membrane, we have BAC and BAX. BAC and BAX are pro-apoptotic gene. So whatever present on the outer membrane is activated and whatever present on the inner membrane is inactivated. So what is in action here is BCL2 and BCL2 is anti-apoptotic. So BCL2 has kept apoptosis in check. Now, whenever there is any form of stress or stimuli, example viruses, irreparable DNA damage, or accumulation of misfolded proteins, they are sensed by the sensors. And I have told you that sensors are present over the cell surface. They will sense any form of stimuli or any form of stress and will inactivate the anti-apoptotic genes. And sensors will activate the pro-apoptotic genes. That's how these pro-apoptotic genes will come on the outer membrane of mitochondria, as you can see in this image. So now these BAC and BACs are activated. They will initiate the apoptosis. How it will initiate the apoptosis? By increasing mitochondrial permeability. And as you all know that there is an enzyme present inside the mitochondria. This enzyme is known as cytochrome C. And due to increase in mitochondrial permeability, this cytochrome C will come out of the mitochondria inside the cytoplasm. When this cytochrome C will come inside the cytoplasm, it will form a complex with apoptotic activating factor 1 and this complex is known as apoptosome. This apoptosome will convert procaspase 9 into caspase 9. So this is the whole mechanism of intrinsic pathway. Here we have inhibitors of apoptosis also inside the cytoplasm of the cell. What these inhibitors of apoptosis will do? They will check conversion of pro-caspase 9 into caspase 9. So we need something that will cause inhibition or inactivation of these inhibitors of apoptosis, and which will inactivate these inhibitors of apoptosis, SMAC dihaloprotein. This SMAC dihaloprotein was also present inside the mitochondria, and because of increase in the mitochondrial permeability, it will come out of the mitochondria into the cytoplasm, and it will inactivate the inhibitors of apoptosis. So if I ask you that SMAC dihaloprotein is pro-apoptotic or anti-apoptotic, then you will answer that SMAC dihaloprotein is pro-apoptotic because it is inactivating the inhibitors of apoptosis. Our next pathway is extrinsic pathway, which is also known as death receptor mediated pathway. We have a few death receptors over the cell surface, and an example of these death receptors are TNF receptor and FAS, which is CD95. Now let us discuss extrinsic pathway in detail. We have a cell, and over the cell surface we have FAS receptors. This FAS ligand will come and join with the FAS receptor. It will attract more of receptors, and ultimately it will form a complex known as FAS associated death domain. This FAS associated death domain will cause conversion of pro-caspase 8 or 10 into caspase 8 or 10. Another thing is we have a protein flip which keeps this conversion of pro-caspase 8 or 10 into caspase 8 or 10 in check. Now let's just discuss apoptosis once again. In apoptosis we have an initiation phase, we have an execution phase. In initiation phase we have intrinsic pathway and we have extrinsic pathway. Now in intrinsic pathway we have caspase 9, in extrinsic pathway we have caspase 8 in lower animals and caspase 10 in humans. In execution phase we have caspases 3, 6 and 7 that will activate endonucleases, and ultimately these endonucleases will cause chromatin condensation. So this is whole of your apoptosis. Now we have discussed apoptosis in brief as well as in details. We have discussed the pathways in details. We have discussed it in brief. Now let's talk about morphology of apoptosis. I have made a table to differentiate between the necrosis and apoptosis. That's how you can easily memorize the morphology of both necrosis and apoptosis. If we talk about the cell size is increased or enlarged in necrosis, but in apoptosis the cell shrinks. Plasma membrane is disrupted in case of necrosis and it remains intact in apoptosis. We have discussed the nuclear changes of necrosis in another video. I will share that link in the link box. You can go to that video and read about necrosis in detail. So the nuclear changes in necrosis are pycnosis, karyorrhexis and karyolysis. Whereas the nuclear changes in apoptosis are fragmentation into nucleosome sized fragments. What about the cellular contents? Because the plasma membrane is disrupted in case of necrosis, the cellular contents will leak out of the cell, but in apoptosis they will remain intact. Talking about the adjacent inflammation, because of the leaking out of these cellular content, this adjacent inflammation is present in necrosis and absent in apoptosis. If we talk about the causes, necrosis is invariably pathologic, but apoptosis can be because of physiologic causes as well as pathologic causes. Now this is again a small summary of apoptosis. Whenever we need elimination of unwanted cells, there is programmed cell death. How it occurs? Firstly, there will be cell shrinkage, there will be formation of some nuclear fragments, and there is formation of these apoptotic bodies. These apoptotic bodies will fall off the cell and send some eat me signals to the phagocytes. And ultimately, there will be phagocytosis of these apoptotic bodies, and there will be no adjacent inflammation in this case. Now, this is something very important for you all, clinico-pathological correlations. Whenever there is any DNA damage by radiation or chemo, tumor suppressor gene P53 comes into act. How? This tumor suppressor gene will arrest the cell cycle and gives time to DNA to get repaired. But what happens if the DNA damage is beyond repair? This tumor suppressor gene will cause apoptosis. It will induce apoptosis. And supposedly, this P53 gene is mutated. What happens then? There will be no apoptosis, and if there is no apoptosis, the damaged DNA will accumulate, and ultimately it may lead to neoplasia. Similarly, we have discussed in the causes of apoptosis that misfolding of proteins may lead to apoptosis. But what if apoptosis fails? These misfolded proteins may get accumulated in our body, and some examples of such diseases are Alzheimer's disease and Huntington's chorea. Here I am going to discuss something about necroptosis. As you can see, necroptosis is comprising of two terms. One is necrosis, another one is apoptosis. So because it is an apoptosis, it is programmed cell death. And why we are calling it as necrosis? Because there is no caspase activation. So that is how it is necrosis plus apoptosis. What are the causes of necroptosis? We have some physiologic causes and pathologic causes. In physiologic causes, we have mammalian growth plate at the ends of bone. In pathologic causes, we have some neurodegenerative disorders and pancreatitis that you need to just mug up. What is the mechanism behind necroptosis? We have these TNF receptors on the cell membrane. Now, tumor necrosis factor will come and bind with the TNF receptors. They will activate RIP complex 1 and 3. What do you understand by this RIP? It is nothing but receptor interacting protein. Now these receptor interacting protein will act upon caspase 8 and they will activate the caspase 8. And if caspase 8 are activated, they will cause apoptosis. But what if these caspase 8 are not activated? They will increase the stress on mitochondria and ultimately there will be free radical generation. Now these free radicals will cause injury to the cell in form of necrosis. So we have cell swelling, cell membrane damage and inflammation, and all these three features are characteristic of necrosis. Now moving on to another newly introduced topic, that is pyroptosis. Pyroptosis, as the name suggests, pyro means fire, and apoptosis is programmed cell death. So it is also a programmed cell death, and where the activation of caspase is caspase 1 or 11. And pyrogens are inflammatory cytokines, mainly interleukin 1. So what you need to remember is caspase 1 and interleukin 1. What happens in this case? Let's talk about the mechanism, then you will remember it. Whenever there is any entry of microbe into our body, it will enter the cell cytoplasm and it will activate inflammasomes. Ultimately, what will happen? There is activation of caspase 1. This caspase 1 will cause the activation of interleukin 1, and interleukin 1 is something which will cause fever and recruitment of leukocytes. So with the name pyroptosis, you just remember caspase 1 and interleukin 1. That is important for you. Sometimes it may be asked as an MCQ in your PG entrance examination as well, that which caspase gets activated in pyroptosis, caspase 1. Which interleukin gets activated in pyroptosis? That is interleukin 1. Now moving on to the morphology. The morphology is similar to necrosis, that is cell swelling, cell membrane damage and inflammation. So that was all about apoptosis. I hope you liked the video. You could understand the pathogenesis, the biochemistry, the morphology and the pathways we have discussed in the video. If you really like the video, please share it with your friends and subscribe to my channel Pathshala by Dr. Rohit Sharma. Thank you.